So before I will start this brief talk, I also would like to say a very warm welcome to all of you, and also thanking you for sharing and celebrating with us this 30th anniversary of the Genebank. So let me introduce you to the Bioversity International Musa Germplasm Transit Center, a very long name for what is known in the banana community and also beyond as the ITC, or the Banana Genebank of Bioversity. As you can see, we're just not named a gene bank, but a transit center, and that's important because our first objective, of course, is to conserve genetic resources, but equally important to our work is that when these accessions and bananas are coming into the gene bank, that they also are sent out again for use. And the fact that we are located in a non-banana growing country, Belgium, where the pests and diseases that are associated with this crop do not occur, this can facilitate these exchanges and the acquisition, the receipt of germplasm, and also its distribution again to other parts of the world. Now, why is banana so important? It was already mentioned. Banana is one of the most important food commodities in the tropics. It's grown in 120 countries. And every year, 145 million tons of bananas are produced. Most of these bananas are produced by small-scale farmers for local consumption. So it are millions of people, millions of farmers who depend for their daily food and income on this crop. And they are actually also the growers of the large diversity of cultivated bananas, the dessert bananas, as well as the cooking types, which comprises more than 500 different varieties. However, it should also be said that 40% of the global production relies on actually only a very few number of varieties, or even one single variety, the Cavendish, which is a dessert banana commercially grown for export. This monoculturing is increasing everywhere in the world, also in the small-scale farmers' fields. And this is actually also a threat to the supply of food, because the production becomes more and more vulnerable to pests, diseases, drought which is even intensified now with climate change. So there is indeed an urgent need that this diversity is again incorporated in farming systems and in production systems. And to underpin this, it's important that this diversity is safely conserved. How is banana diversity conserved? Well, as we already heard, banana is a seedless crop. It is vegetatively propagated through shoots. Therefore, the traditional way to conserve genetic resources of banana is in field gene banks. Field gene banks where the plants are grown in full size. But these plants are exposed to pests, diseases again, and also to adverse weather conditions, so they are vulnerable. That's why it's important that they are backed up in a more secure way, for instance, through in vitro conservation. In vitro gene banks are repositories for genetic resources in the lab, where they are maintained under the form of small plants in glass test tubes. We will see a very nice example of this conservation approach later today during our visit to our facilities, namely the Banana Gene Bank managed by Bioversity International. So Bioversity International here in Leuven manages the largest collection of banana in the world. It comprises more than 1,500 different samples or accessions, as we call it. The collection was established 30 years ago, more or less, in 1985 at KU Leuven. And in 1994, it became also an in-trust collection under the auspices of the Food and Agriculture Organization of the United Nations. Since then, we have the commitment to preserve these genetic resources for the long term and also to keep them accessible for the world community. 10 years later, when the International Treaty on Plant Genetic Resources for Food and Agriculture came into force, The collection was placed in the multilateral system of access and benefit sharing. This created the legal framework for the exchange and use of this germplasm. So now, the home of this collection is KU Leuven. So we are very grateful to the university for sharing with us the facilities, excellent laboratories, greenhouses, their services, which are really essential to operate this gene bank. But more importantly even is the scientific collaboration, the scientific partnership with this university. Because for more than 20 years, the university has been conducting research on conservation technologies, which resulted in the methods that are now daily used to conserve and to manage our gene bank. These are, for instance, the cryopreservation techniques that were developed and that we will be showing you later this afternoon as well. Today, the research focuses more on creating value, adding value to our collection through studies on, for instance, the trait of our accessions in the gene bank. And they are particularly focusing on broth. And this work will also be shown to you when we visit the greenhouses. So from the early days, the gene bank has been acquiring very actively new materials to become what it is today. We have been introducing samples from 60 donor sources in more than 30, nearly 40 countries. These are mainly duplicates from the major field collections around the world, but also new materials that are discovered still in the tropics and are added to this valuable collection. Here, for instance, we see two taxonomists on collecting mission in Indonesia with their latest discovery, which also soon will be included in the gene bank here in Leuven. So today, the collection is the largest repository for genetic resources of banana in the world. We have 1,536 different bananas. Most of them are cultivated bananas, 75%, a wide range of eatable dessert and cooking types. As you can see here, a lady in the Pacific with her Pacific banana plantain. 15% are actually wild materials and 10% are improved varieties. These are superior banana varieties which have been produced by breeding programs of banana worldwide and which are of great interest for users. All this diversity is safely conserved in the active in vitro collection here in the lab. So this collection is actually a very small room which holds 30,000 tubes containing bananas, small plants of bananas. So I will be happy to show you the method of this collection and how we operate it when we visit our facilities. This is also the collection that gives direct access for users. We also have a second collection, which is called our cryopreserved base collection, and which uses the cryopreservation technologies to be established. So here we are actually freezing the little pieces of bananas into liquid nitrogen at ultra-low temperature. So this collection is actually conserving the germplasm in a frozen state. And this allows us to conserve it on the long term in a safe manner, we can say in perpetuity. And although this collection is not yet complete, it holds 2/3 of the total diversity present in the gene bank. It is the world's largest cryopreserved collection for banana. And we have a third collection, a leaf tissue collection. So to better serve our users, we also established this collection, which consists of leaf tissues harvested from greenhouse plants. And the aim is actually to preserve the DNA of the germplasm in the collection. But it also helps us to facilitate the exchange of genetic materials with, for instance, scientists involved in molecular research. So this is an important complementary approach that we are using to conserve genetic resources in the lab. You see, as you can see, a lot of efforts go into the conservation work, but next to that, it's also very important for us that the germplasm in the collections is being used, so that users, like researchers, have access to it, to get new knowledge, to gain new knowledge about the crop, but also farmers, that they can directly benefit from the wealth of diversity that is present in this gene bank. So over the years, we have been obtaining a very prominent position in the world in distributing germplasm. And this was thanks to a system of safe exchange of plant materials that we established. And the basic principle of this is that only healthy materials are given out to users. So substantial efforts also go in testing our banana materials to ensure that they are clean from any pest or disease. Samples can be easily obtained from our gene bank. It's just with a few clicks that users can request germplasm. And this is thanks to the system called Musa Germplasm Information System, a database system on genetic resources of banana that was developed by Bioversity, and that holds information, descriptive information on each accession in the gene bank. So based on this information, users can select directly online germplasm and request it with a few clicks. And the germplasm distribution activity that started 30 years ago is impressive, because we have been supplying samples to more than 300 users, particularly users in developing countries in 110 countries. 3 to 4 accessions are shipped every day from this collection to another place, to some place in the world. This germplasm that we distribute is also intensively used. Most of our material is being used for research to gain new knowledge about the crop, but also a substantial amount of germplasm that we distribute is going directly into cultivation, adopted by farmers. Germplasm is also used for evaluation trials to get a better understanding of the agronomic traits of the materials that we hold in the collection, to characterize them, to understand the diversity in the collection, and a lot of materials are also sent back to field collections to restore them and to broaden the diversity in the national programs where in the countries where bananas are grown. And last but not least, we also support breeding programs around the world, breeding programs on banana, by supplying them samples of particularly wild materials, because these harbor the genes that are of interest to produce, to breed superior bananas. To conclude, I just would like also to acknowledge our partners and to express our thanks to these partners for their support, because without their continuous support, it would not have been possible to operate this gene bank in the past 30 years. So therefore, thanks, and also thanks for listening.